The process of cellular respiration occurs in both the cytoplasm and the mitochondria, and it happens in plants and animals. It can happen in anybody that has a mitochondria. It's a 3-step process, although there are some littler kind of linking steps in there. The first step is called glycolysis, and that happens in the cytoplasm. The next two, the citric acid cycle and the oxidative phosphorylation portion, both occur in the mitochondria. This whole process turns one 6-carbon glucose molecule into approximately 28 to 32 ATPs. Sometimes you see numbers as high as 38, but we're going to stick with 28 to 32 just to keep it simple. So let's talk about that first step, glycolysis. So again, we're out in the cytoplasm at this point in time, and we start with one 6-carbon glucose molecule. We attach 2 different phosphates to it, which gives it the energy to break apart into smaller component pieces, and you end up with a lot of intermediate forms. We're just going to stick with kind of the basics, ins and outs. So as you go through those intermediate forms of glycolysis, as you're breaking some of those bonds, you end up removing electrons and hydrogen ions, and you move them onto cellular batteries, known as NADH when they're charged. When they're not charged, you haven't used the battery yet, we call it NAD+. We'll get to those batteries later. Just know that as we break these bonds, we release electrons and hydrogen ions, and we capture them for use later. The other broken bonds are also used to make 4-ATP. The component parts, the ADP and the phosphorus, are just consistently floating around in the cells, so we use the energy to push those 2 items together. So in glycolysis, we have a net gain of 2 ATPs because we had to use 2 to kind of kick it off. At the end of this glycolysis step, your 6-carbon molecule that you started with has been turned into 2 3-carbon molecules that we call pyruvate. The next step of the process is to alter those pyruvates we just made. We've entered now into the mitochondria, and we're going to use some enzymes called acetyl-CoA to remove yet another carbon. We've broken a bond, so let's harness that energy to charge another NADH battery. That pyruvic acid gets a name change because we've changed its chemical structure. We've removed the carbon and added that CoA enzyme. So now we're going to call it acetyl-CoA. We have to go through this twice because we had 2 pyruvates from the previous step. So in the creation of acetyl-CoA from pyruvate, we get 2 NADH batteries and we lose 2 more carbons. You end up just breathing them out as CO2. This whole process is known as the grooming of the pyruvates. We had to get them chemically ready to enter into the next step, which is the citric acid cycle. The citric acid cycle consists of 6 steps, but we're going to kind of clump them together for ease of studying. You'll get more specific as you take higher level bio classes. So in step 1, the acetyl-CoA kickstarts the process. That CoA actually immediately leaves. There are 2 carbons left over, and they bond with a 4-carbon molecule called oxaloacetate that's been hanging out in the mitochondria for this exact purpose. In steps 2 through 3, we go through a lot of redox reactions, or reduction oxidation reactions, simply meaning we're going to move electrons from one molecule to the next. Our 6-carbon molecule we just made is broken down, and every time we break a bond, we collect either the charged electrons to form some more of those cellular batteries, or we just make a molecule of ATP. All of those popped off carbons are breathed out as CO2, just like before. In steps 4 through 6, we simply undergo more of those redox reactions where we're kind of reshuffling some electrons. We keep breaking bonds, kicking out carbon, and charging batteries. In this instance, it's important to note that some of the batteries have different names. 2 of the batteries that we're going to charge are called FADH2. They function a little bit differently when you get into the specifics, but we're just going to know that they're a little less effective at transferring energy. So in summation, in the citric acid cycle, we break down all of the bonds. At the end of the citric acid cycle, no carbon is left over. It's all been breathed out as CO2. We have, however, made 2 ATPs, 6 NADHs, and 2 FADH2s. So we keep charging all of these batteries. Well, what are we using them for? We're going to use them to create an electrochemical gradient. That's the last step, which exists in 2 parts. The first step, called the electron transport chain, uses all of our batteries. The NADHs and FADH2s donate their electrons to proteins that span the inner mitochondrial membrane. As they were picking up electrons, they were also gathering those H+ ions from the broken bonds. The donated electrons give the proteins just enough energy to pump all of the hydrogens between the two different membranes in the mitochondria. Once a gradient is large enough, a molecule called ATP synthase opens up. This is the second step in the process, and it's called chemiosmosis. The ATP synthase works as a rotor, and it spins quickly from the rush of hydrogen flowing through it, attempting to balance out that gradient that we just created. This spinning physically forces ADP and phosphorus together to make molecules of ATP. We can create from 28 to 32 ATPs from this gradient, created by just one hydrogen. Here's that simplified process again. Now there's a lot more to it, and I look forward to the day that you'll get to learn all of the kind of intricacies, but for the sake of our class, let's keep it at this particular level. In the cytoplasm, you have glucose, which with the help of two ATPs is reorganized and broken down into two pyruvates, which are both 3-carbon molecules. In that process, you gather enough hydrogen and free electrons to charge 2 NADH batteries. You also make 2 ATPs. The pyruvates enter into their grooming phase. They lose a carbon and pick up a CoA enzyme. In this process, we also charge 2 NADH batteries. Our new molecule is known as acetyl-CoA. It's capable of entering the citric acid cycle. It undergoes many redox reactions, and all of the carbons that used to exist in the molecule are broken down and breathed out as CO2. You charge 6 NADH batteries, 2 FADH2 batteries, and create 2 ATPs. All of those batteries are used in the electron transport chain, seen here. The batteries donate their electrons to allow a physical change to occur in some proteins. Those proteins pump the stolen hydrogens into the space between the two membranes that exist in the mitochondria. I would like to point out on this slide that you see oxygen at the very end. We have all those electrons that are being passed from protein to protein. Once they've flowed down through the system, though, you need to do something with those electrons. You don't just want them floating around in your cell randomly bonding. That could actually be dangerous. We need to do something with all of those hydrogens as well. What our cells do are attach the free hydrogens and the free electrons onto an oxygen molecule, and we make H2O. In fact, you make about 20% of the water that your body needs every day by capturing free hydrogens and electrons on oxygen. This is why you breathe. That oxygen is really important. One more time, here's an overview slide. A lot of students find this particular picture quite helpful, although it's not in your version of the book, so I've provided it here. Pause the video and see if you can't walk yourself through it. Cellular respiration is dependent on oxygen. It's aerobic respiration. There are some instances where there isn't any oxygen or there's simply not enough. One example of what your body can do when you don't have enough oxygen is lactic acid fermentation. It's really common in muscle use when we're exhausted. The glucose goes through the process of glycolysis and becomes 2 pyruvates. But because there isn't enough oxygen to capture all those free electrons and hydrogens, it's dangerous to push all of this material through cellular respiration. So instead, our body chooses to ferment those 2 pyruvates and turn them into lactic acid. We can simply charge electron batteries in this instance and use them to keep breaking down glucose, and at least we get those 2 ATPs. It's better than nothing. Another example is alcohol fermentation, which you might be familiar with if you're interested in brewing beer or making wine. We changed pyruvic acid into CO2 and acetaldehyde. The acetaldehyde then undergoes reactions to become ethanol. In this instance, you can see that we actually have to use a battery for this process, so it requires a lot of sugar, which is why you need a lot of sugar when you're brewing alcohol. Brewers and winemakers think about all of these components and make machines that deal with it. The fermentation of grape juice into wine produces a lot of CO2 as a byproduct. Fermentation tanks have special valves that allow the CO2 gases to be released so they don't burst. Not all systems use this exact same process. Some of them them can replace the needs for carbon or oxygen with hydrogen sulfide instead. What you end up seeing are releases of hydrogen sulfide gas. In the instance of these particular bacteria that are decomposing algae using a process with hydrogen sulfide, you end up with this kind of neat green color in the water. Here's this process laid out again, considering some of the other things that you probably consume in your diet. Carbohydrates and some amino acids in a molecule called glycerol can enter glycolysis at the very beginning of the process, and we can create our 20H32 ATPs from it. If you're only eating fatty acids and some amino acids, your body actually skips the glycolysis process and moves straight into pyruvate oxidation. Other amino acids have to start with the citric acid cycle and move on to oxidative phosphorylation. Some people think, well, if you're not taking in those extra calories, isn't that a good thing? Not necessarily, because you're missing out on all of those batteries, and therefore your end process is lesser than it could be if you were eating foods that provide better energy. With that, we end our study of Chapter 4. You should make sure to read the book, take notes in your own words, and study. Watch some videos from a few different perspectives. This is your first really tough topic, and you'll probably find it helpful to hear it explained by other people. Once you're done, take a crack at your homework.